In this video, we will discuss about this ARHGF1 gene knockdown results in the reduced metastasis, and it reduces metastasis by down-regulating T cell functions. So these genes informations, they come up from the bioinformatic analysis of the in vivo samples that are done by the researchers, and out of that they wanted to check the detailed impact of this ARHGF1 knockdown. So when they use this ARHGF1 knockdown mice and injected them with the cancer cells, what they are able to see reduced metastasis in this ARGF1 knockdown that can be seen in this panel A, and these things are quantified in the panel B, and the metastasis that they have checked is on the lungs, and we can see that the metastatic nodes or nodulus are lower in the case where ARFGE1 is knocked down. And when they check the metastasis for area per lungs, that is also reduced in the case of this ARFGE1 knockdown. Then they check the metastasis on the liver, as this ARGF1 functions is not only limited to the lungs, and in the liver also they are able to find out that the metastasis is reduced when they use ARFGF1 knockdown mice. Thereafter, they wanted to analyze this metastasis. So how they did that is that they take this mouse model that has this MMTV PYMT, and one of the mice has this ARGF1 knockdown and one of the mice has this ARGF1 wild type, and then they analyze the samples in both the conditions, and this is the wild type lungs looks like, and this is the ARFGF1 knockdown lungs looks like. There are no black dot. Black dot represents the metastasis of the cancers into the lung area. So for this C part, I was forgot to explain that we can see that the metastasis is not only reduced in the lungs but is also reduced in the liver area, but there is not much impact they have seen in the primary tumor mass, and however the total metastatic tumors is reduced. And thereafter they create a cell lines where they induce them with these BM cells, and the mice models has either ARFGF1 knockdown or wild type. This is not a cell line, this is the in vivo studies. And then they check the nodes in the per lungs area, nodes per lung pair, and they are able to see, so in the ARFGF1 knockdown the signals are lower. So this further substantiate their point that ARFGF1 plays a role towards metastasis of the cancers. When they check the pathways or the genes that are upregulated, they are able to see most of the immune suppressive genes are upregulated, such as CCL4, NL, NKG7, and the other genes that are shown in this ARHGF1 knockdowns. Now the hypothesis that this upregulation of the genes might play a role towards the suppressions of the metastasis. So they create a reporters of this ARHGF1 that can target NK cells, macrophages, and T cells separately, and then they co-culture this these cells with the NK cells, macrophages, and T cells. And what they are able to find out is that in the case of NK cells, there is no decrease in the metastasis. In the macrophages also, the metastasis decrease is not observed, but in the T cells, they can see the metastasis in the lungs area is decreased. The other two IJ are also checked for the lung pair, but for the T cells, they are able to see decrease in the metastasis. And here, what they are able to conclude that it's the ARFGF1 knockdown might results ARFGF1 knockdown might results in the immunosuppressive upregulation of the genes that helps these T cells to perform its functions and reduce the metastasis. and and in the conditions where ARHGF1 is present, that it will prevent the immunosuppressive upregulation of the genes that plays a role towards immunosuppressions, and thereby the T cells won't be able to reduce metastasis. These things they have confirmed in the mice models thereafter, where they check this wild type and the ARHGF1 knockdown mice, and they can see that the metastasis is far lower in the case of ARHGF1 knockdown compared to the wild type. So this substantiate their previous findings that ARHGF1 knockdown reduces metastasis in the cancer cells, and the mechanism that they are expecting is that it will suppress these T cells functions. I hope you understood these experimental results to some extent. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description, or you can also write down the comments. I will try to explain that part. That's all for this video. Thank you for your time.